I'm delighted to welcome Dr. Andy Maloney from the Cambridge Crystallographic Data Centre, CCDC, the long-standing Tier 2 partner within CMAC and involved in a number of the programmes. And it's great to get their perspective and some of the opportunities in this area. So Andy is going to lead on our keynote presentation with how far will a crystal take you, which I'm looking forward to getting an answer to that.
Thanks very much, Alistair. So when Alistair asked me to give this, he wanted me to talk about opportunities, perspectives on digital design and pharmaceutical development, with really a focus on collaboration. And so it's not the only pun that I've tried to put into this presentation, but it's probably one of the better ones. Sorry about that for the title. So I'm Andy. I'm from the CCDC, the Cambridge Crystallographic Data Centre. Most of you probably have heard of us, but for those of you that don't, we're a charity based in Cambridge that's really there for the advancement of chemistry and crystallography for the public benefit. We do lots of different things with that remit though. So primarily we're an international repository of 3D structural information, so crystal structure data. We're also a collaborative research organisation. We provide knowledge-based services. That's a lot of the contribution that we make to CMAC and a number of our partners. We develop scientific software that goes along with the data and to use the database. And we're quite focused on education and outreach as well, to ensure that scientists throughout the world are able to use the resource. And that resource in question is the Cambridge Structural Database, or the CSD. So the CSD was established in 1965, so we're approaching our 60th year now, and from sort of fairly humble beginnings of a few hundred structures, we've recently surpassed 1.25 million. So that's quite a lot of data, and that data comes from a lot of different sources as well. So you can see on the slide of the slide there, it's published structures from the scientific literature, and so this comes from a whole host of places. Journal articles, for instance, but you can also publish structures directly through the CSD. We get structures from patents, from university repositories, even from theses, even some sort of handwritten old structures from sort of the early days of crystallography find their way in there. And we've got a team that makes this a valuable resource for the community by enriching and annotating it. So really structural chemistry goes into that there. And we are a core trust seal repository as well. That's important if you care about data standards, and maybe a bit more about that later. But there's an awful lot of information in the database as well. So I said we've got over 1.5 million structures, and that's a huge number of 3D coordinates of atoms. And that tells you an awful lot about bond lengths, about valence angles, torsion angles, ring geometries. And so an awful lot of information in there about structural chemistry. And that structural chemistry comes in a lot of different guises as well. So this is the makeup of the CSD, and you can see a few interesting stats from this. So it's a roughly 50-50 split of organic and metal organic systems. I really care most about the organic systems, as do probably most of the people in this room as well, but there's an awful lot of transition metal chemistry in there as well. Most of it's not polymeric, most of it's single component, but we've got quite a variety of different structures in here. So drugs is probably what we're going to focus on for this talk, but there's lots of other interesting functional materials in there into the database. And we do try to link it with as much information, with as much metadata, and with access to as many other repositories as we can. So we do have links with some other databases that you can see on the slide there. We have an awful lot of additional experimental information as well. But what we really care about here, what we're thinking about is drugs and medicines. And so on this slide here, all of the boxes that you see in green are from the top 200 drugs by retail sales, and they're structures that are in the CSD. So that data's there. If you want it, you can come and get it from us. The grey ones are sort of those big wobbly macromolecule biological things that are quite scary, so we'll ignore them for the purpose of this talk. And the white ones are the ones that aren't there yet. So if any of those are yours, you know where to send them. But with all that information about drug molecules and all that information about structural chemistry, we can put together knowledge-based solutions, so some software to really help drill into these things. And so some of you will be more familiar with others of these. We've got a stand outside if you want to really know more about it. But these are focused on making the most use of the data in the CSD to answer sort of real questions about designing new drug molecules all the way to thinking about particle behavior and how predictive you can be using sort of fairly high-end computational techniques as well. Really, for the purposes of this talk, though, I'm going to focus on CSD materials, CSD particle, those suites. We'll maybe touch a little bit on CSD theory at the end if we've got a little bit of time. But this is how we can take the CSD as a resource to think about digital design, digital transformation in the pharmaceutical industry. And this has been a long journey, right? This didn't happen very quickly. And so here is my very incomplete and very biased timeline of what's happened in the last 25 odd years. To how we got to where we are today. And so I don't need to introduce what happened in 1998 to this crowd, I don't think. But ritonavir happened. So uncontrolled polymorphism in a pharmaceutical product that was on the shelf, right? That's a big problem. And people wanted to know how we could stop that happening again. And so there was an awful lot of interest from across the pharmaceutical industry to thinking about predictive ways. How could you have recognized that a more stable set of hydrogen bonds would form after a given amount of time in a drug product? And so, like I said, lots of interest from the industry. From a CCDC perspective, that really kicked off in 2002, when Pfizer invested in the CCDC and in the University of Cambridge to develop the Pfizer Institute for Pharmaceutical Materials Science. And that really put an awful lot of the sort of fundamental science that we're going to talk about today into our tools, into our workflows, and really sort of made us think about the CSD being the resource for the pharmaceutical industry that it is. More people got involved and got interested in this, and so 2008 saw the establishment of our Crystal Form Consortium. So this is a group of pharmaceutical partners, and a lot of you are in the room today, that make use of CCDC tools and our knowledge to help their digital development workflows. Something happened in 2011, but we won't dwell on that for too much. But CMAC got established, and that sort of really gave us a means to join into a bigger community and start to think a little bit more about pharmaceutical manufacturing and how we can take a structural chemistry approach to that. And that was really benefited from the ADOPT project in 2015. So this was us really sort of starting to think about digital design and actually putting into action some of the ideas and some of the workflows that we'd had to go from knowing about a crystal structure to knowing about a particle and maybe even its downstream properties. Like Alistair mentioned, as Tier 2 partners, we've been part of a number of different collaborations that have involved CMAC. So Articular was a really interesting one with Blair that we did. DDAP was a big collaboration as well. I realised that when I was putting together these slides that we never actually came up with a logo for DDAP. So I did that myself, and I think you'll see that graphic design is obviously my calling. We've had a lot of collaborations with colleagues at the University of Leeds, both part of CIAC and outside of that as well. And more recently, we're really starting to get to grips with the data-focused approaches through DM squared and DDMAP and how we can contribute there. What Michael's doing with amorphous systems using crystallographic data is really, really quite fascinating, and I'm keen to see how far we can take that. So that's where we got to, and from the end of that, we got some workflows to do that. And so we split these workflows into sort of two different parts. We call the approach that we take by using crystal structures to understand the data from crystal structures to understand these problems, structural informatics. And you can split that into two halves, really. So we've got solid form informatics and particle informatics. I'm going to talk you through a bit about what's in them, how they came to be, and the collaborations that put them there on the way. But really what this does is it gives us a platform, a way of thinking that can take us from thinking about a molecule and its context in the solid state through to thinking about a particle, its properties, and ultimately a drug product. And so many of you will have seen this next slide before, but in terms of, this is a nice way of explaining structural informatics. So by show of hands in the room, which one of these is the stable wall? Put your hand up if you think it's wall A. Good. Now put your hand up if you think it's wall B. Good. Who's actually built a wall? Okay, but that's fine, right? Because you have looked at the database of walls throughout the world and you have made a number of observations. That all these structures have motif A in them and none of these structures have fallen down yet. So there's something inherently stabilizing about this motif A, and because we see that again and again and again, there's an indication of stability from that particular motif, that arrangement of bricks. And we don't really care about bricks, right? We care about molecules in crystal structures. And so what we look at is we look at data trends across the CSD to understand the different characteristics that influence stability in the solid state. And so we can think about the conformation of molecules, but we can also think about the intermolecular interactions, hydrogen bonds, non-hydrogen bonding interactions, and how they all contribute to the stability and the relative stabilities of different polymorphs as we see them. And so we can derive knowledge from the CSD. So that's how we establish these informatics workflows. And so a couple of simple knowledge bases that have been around for quite a long time, if you look at those references on the bottom of the screen there, focus on looking at that. So we have MOGL that's focused on intramolecular geometry, so understanding valence angles, torsions particularly, things like conformational polymorphism, we can drill down into this. And ISOSTAR lets us think about intermolecular interactions, and so how we can see these geometric distributions across structures in the database. And so the way that MOGL works, sort of very, very briefly, is that we take a particular bit of chemical functionality, we can draw that out. You probably can't see it, but trust me, all these connected rings are slightly different in the heteroatoms and the substituents that they have on different positions. And we can look across the CSD for similar structures and look at the geometries of those similar structures. And we can start to build up these histograms, these distributions of observations that we make from that data. And when we compare that to all right-ish computational methods, dihedral scans, we can see that it actually maps up pretty well. You know, if you look at the big purple line, the one with the big peak in it, if you look at the MOGL distribution, so the distribution of geometries from the CSD there, you can see that you have population in that distribution at either end at 0 and at 180 degrees, and you've got nothing in the middle, right? You've got this big barrier to rotation there. And so what we're seeing is we get this high-end computational information from the database. The other, I'm happy to come back to this slide if anybody's really, really interested in it. But the other histograms and scans show the same sorts of things. In terms of the intermolecular interactions themselves, these are a little bit harder to think about sometimes. We tend to focus on hydrogen bonding because it's directional and it's sort of simple to think about. But the nature of hydrogen bonding and those geometries can be very variable in the solid state. And so if we want to think about drilling down into what is going on here in terms of the CSD, we can use ISOSTAR. And so that helps us to extract different intermolecular interactions. And so if we focus on an amide and an amine, for instance, as we can see here, we can look at two different structures on from the CSD, and we can see that the nature of the geometry of those interactions is a little bit different. And so we can superimpose those on top of each other. And if we do that for all the structures in the CSD, we can start to build up this density of interactions. And what you can see here is that your amines interacting with your lone pairs on your carbonate, right? That's high school chemistry sort of stuff. But we can see that and we can prove that from the data. And because we've got an awful lot of data to build that from, we can start to build up densities. And so we can well, maybe you can't see it very well on the slide there, but we've got these really, really obvious peaks there around about where we'd expect to see the lone pairs in the carbon and oxygen. And so we don't just have to focus on individual intermolecular interactions, we can sort of combine these together. And we can, when we break down a molecule into different fragments and look at the interactions for each fragment, then put them all back together again and think about the sterics of what's going on. So if we sort of start to, when we sort of start to build them up and think about different bits of the molecule blocking out different other bits, we start to build up a picture of how that molecule might like to interact in the solid state. And so we can take that molecule and we can put it back into the crystal structure effectively that we're interested in. And we can look at the directionality and the geometries of the intermolecular interactions that we form, and we can start to understand if from a geometrical perspective, they're going to be very well satisfied. Now, if you remember sort of this construction a little bit later on, it will become a little bit more important. It's how we're starting to use it to think about more downstream processes as well. But these different informatics tools, these different approaches, this solid form informatics, we've put together into this health check methodology, which we've really put together with colleagues from the Crystal Form Consortium that have enabled us to think about really refining these and putting together a workflow where we can start to think from inception of a new molecule. As soon as you get at the first crystal structure, what can you do with it and how can you use that to understand pharmaceutical development? So by understanding the geometries, understanding the intermolecular interactions, doing some really, I don't have time to go into it today, but doing some sort of some logistic regression, building some clever models to think about probabilities of hydrogen bonds forming, we can sort of really start to make assessments about the relative stability of the different solid forms. Do you have the most thermodynamically stable polymer in front of you, or is there something somewhere that if you find it, it's gonna maybe cause you some problems downstream? And so this is one of the approaches that we take with the information that's in the CSD, but we can actually look at more of what's in the CSD to understand what drugs do in solids a little bit more. And so this is a collaboration that we did as part of the ADOPT project, which focused on making a drug subset of the CSD itself. And so that lets us think about the diversity of different solid forms that we see for drug molecules in the solid state. So we get quite a lot of them just form pure compounds, and that's nice. You get an awful lot of salts, as you might expect. You get an awful lot of salt hydrates. We probably expect that from what we would expect from our understanding of what we know about salts. Salts tend to form hydrates, right? And we see that in the data as well. Sometimes you form a salt-solvate hydrate co-crystal, and then I think you probably go into a dark room for a bit if you're one of the 16 people that's found one of them. So there's an awful lot of diversity in your solid form landscapes of these things as well. And we can sort of start to drill down to understand what's in the CSD and what is in a more modern pharmaceutical database, shall we say. And so as part of this collaboration that we did through the ADOPT project, we were able to compare the drug subset of the CSD with the organic subset of the CSD and with the in-house databases of AstraZeneca and Pfizer. And you can sort of see the distribution there. And this is sort of like classic Lipinski sort of molecular obesity sort of thing. Molecules are getting bigger, right? We need to be able to understand that and deal with it. And you can see that in the chart here. I should thank the collaborators that did this. So this was Helen Blade and Stefan Taylor at AstraZeneca. Kevin Back at Pfizer did all the work, and Bob Doherty took all the credits, and that's probably pretty standard practice, to be honest. But again, we can sort of see the trends of how things are a little bit different in in modern pharmaceuticals and modern pharmaceutical databases compared to what we see in the CSD. And you'd expect that from the sort of lag thing. You're not taking your development molecules and throwing them straight into the database so that everybody in the world can look at them, right? So we need to make sure that we can understand what this means in terms of some of the trends that we observed and the way that we explore and understand different problems. And so molecular weight's an interesting one because we know that molecules are getting bigger, right? But what does that mean in terms of their solid forms? Well, we can look at that, and we can look at the solid form diversity as we increase molecular weight. And there's a couple of interesting things to take away from this graph. And they're not really trends, but it's interesting observations, I think, that we see. As our molecular weight goes up, our instance of forming a co-crystal goes down. Right. So what does that actually mean for the way we're able to deliver larger drugs as complicated multi-component systems? Right? As we increase molecular weight, our instance of solvation goes up. So bigger molecules are more likely to form solvates, right? And solvates can be very challenging to deal with when it comes to developing a new molecule. So we've got this. As drugs get bigger, we're going to have more complicated issues with developing new solid forms, and we can sort of see that from the trends that we see in the database here. Now, one of the benefits of collaborating with CMAC is that if we want to, we can go big and think about sort of bigger data approaches, and we're really set up to do this quite nicely. And so, looking into solvates in a little bit more detail, we had a PhD student with CMAC a couple of years ago. So Laura successfully defended her thesis last year, and her project was focused on thinking about solvates in the CSD. How predictive can we be, and how can we understand them in terms of their behaviour? And so there's a couple of graphs that I've pilfered from her thesis, to be honest, that we can look at. So different solvents that she looked at and the way they behave in terms of the number of channels that they form in a structure. And so the formation of channels is interesting because as solvent molecules can move through channels, that can have an effect on the stability. And so you can sort of see that things tend not to form that many channels a lot of the time. Some do a bit, some don't a bit. If you're forming more than 4 channels, it's probably a bit of a red flag, and you should really work out what's going on there. But DMF does sort of weird stuff. That actually quite likes to form channels. So there's something interesting about DMF compared to all the other solvents that we looked at, and it behaves slightly differently. And we can look at the nature of the interactions it makes and the way that it forms channels. And so this is a slightly confusing graph on the right-hand side. It always takes me a little bit of time to think about it, and hopefully you can see the key on it. But what it's showing you is when solvents form of all of the solvents in our data sets, what percentage of that data set for each solvent either forms hydrogen bonds or doesn't form hydrogen bonds, and forms channels and doesn't form channels. And so we can see, if we look at DMF as an example again, we form quite a lot of channels. We know that from the first chart, and we form quite a lot of hydrogen bonds in those channels. So it gives us a little bit more information to think about what's going on in these solvated structures. And it was a really interesting project for sort of probing that a little bit deeper from a sort of data science point of view. I want to think a little bit now about particle informatics, and so this is the newer thing that we've started doing more recently. So solid form informatics focuses on thinking about the relationship between the molecule in the solid state, and thinking about the nature of the solid form. So thinking about thinking about polymorphism, thinking about multi-component forms, etc. Particle informatics takes that a little bit further to the next step, and I think this is really where we're starting to see things about how we can take the knowledge that's in the database and translate that into thinking a bit more about how the relationship between crystal structure and sort of late phase development, more into manufacturing as well. And so we can start to think about the relationship between crystal structure and the shape of the particles that we make and the properties of those particles as well. And so that's led us to CSD Particle, which is our newest suite of software that we have. This has been done really through a whole host of collaborations. So it started off in the ADOPT project, and we were sort of really got to do a bit of cool research at CCDC to think about crystal structures and think about them in different ways. That let us think about how we can start to think about bulk properties. So what are the important structural features that might lead to interesting mechanical properties? And so if we can identify and analyze slip planes or potential slip planes in a crystal structure, that's a really good starting point for thinking about how a molecule or how a crystal structure is going to behave when we start to compress it and we try to turn it into a tablet. Through a very long-standing collaboration now with the University of Leeds, we've been working on new ways to calculate morphologies, so we can go from our understanding of our crystal structure to predicting the shape of the particles that we might form for those crystals. And really through a little bit into the ADOPT, but mainly through the DDAP project, we were really able to refine this way of thinking about crystal structures and thinking about surfaces. Now, those surfaces can be your internal surfaces that you might find at the slip planes, in slip planes when you look at your structure, but more commonly, there'll be the surfaces on your morphology that you're thinking about. And so there's ways that we can visualize and analyze these on a molecular level, really, to get a little bit of an idea about what's going to be going on in your system and how we can use that information in the CSD to think about it. And so from our crystal structure, we can sort of start to generate these surfaces. So what you can see on the sort of bluey yellowy one is some sort of physical descriptors of your surface. Now that's showing you your topology or your roughness of that surface effectively. Your yellow bits are sort of raised out of the surface, your blue bits are sort of depressed into the surface. You can sort of see you've got these ridges that are running across that there. Now we can drill into that a little bit more deeply and start to think about physical descriptors such as the surface roughness of that, and actually start to put numbers on these as well. We can also look at chemical descriptors as well. So the top right there shows you the surface chemistry on the surface. So red bits are where we have hydrogen bond acceptors on the surface, blue bits are where we have hydrogen bond donors, and your orangey browny bits are where we have aromatic groups on the surface. So very quickly we can visualize the surface chemistry, the surface roughness of our particles that we're interested in. These full interaction maps that I talked about earlier as well, we can calculate them over the surface. And so we're actually starting to do some really interesting work. Now you can see an example of what that looks like in the bottom right-hand corner spinning around. But we're able to start to think about that in terms of what effect an impurity is going to have on a surface, what effect a solvent is going to have on the surface. And can we start to answer questions about crystal growth and really improve on our morphology models as well. And so we can bring that all together by thinking about these individual surfaces and being able to calculate a good morphology for our system. We can get a sort of surface weighted average of what's going on in terms of our physical and chemical descriptors and come up with these sort of aggregated particle properties as well. I want to go into the slip planes bit in a little bit more detail. I think I'm all right for time. because that's a focus of an interesting collaboration that shows the other end of what we can do at CMAC. And so this approach to thinking about slip planes we developed during the ADOPT project, and we sort of took what Calvin's son in the University of Minnesota had done by sort of looking at crystal structures for a very long time and automated it. And so basically what we do to identify a potential slip plane in a crystal structure is we take a slab of molecules and we split that slab into two different slabs. So we have a blue on the top and a red on the bottom, and then we draw a little line through the middle of those two slabs. And so we do that across all of the Miller planes that we have in our crystal structure. So basically, we're just looking at lots of different planes through our crystal structure, and we measure the degree of interpenetration from one slab into the other slab. And so if we've got a nice gap between these two slabs, that's a nice potential slip plane. If we've got something like this where it could maybe sort of slip like a bit of corrugated steel or something like that, that's maybe a slip plane. That's why I've got to go a little bit further into our thinking about it. But this approach is quite interesting. It's very, very quick. And it tells you an awful lot about what's going on. It's very useful for comparing the relative behavior of different polymorphs. And so sulfamerazine is quite an interesting example of that. So we've got two forms of sulfamerazine that we're interested in, form 1 and form 2. Form 1 forms much stronger tablets at a range of compaction pressures than form 2 does, and so this can actually be related back to you don't even need to put the guiding lines on for this really. If we look at form 1, there's a quite an obvious slip plane between the layers of molecules that we have that just isn't there in form 2 in the same way. And so it's this structural feature that gives us a little bit of insight into what's going on there in terms of the mechanical properties, in terms of what we can see in these systems. I mentioned another collaboration with CMAC, and this is still an ongoing PhD. Julia's not here today; she's at Eli Lilly in the States doing a placement just now. But rather than thinking about a big data approach to it, we're doing some really targeted, I think, fascinating experiments into high and medium pressure X-ray crystallographic studies of pharmaceuticals. And so what you can see on the right is afloxacin being squished, right? So we're sort of thinking about what's happening when we're actually tableting things, but we're thinking about that on a molecular level by looking at crystal structures. And that's really helping us start to understand the link between these structural features and the mechanical properties that we see. So you can sort of see, as that's getting squished there, we can start to think about the slip planes and the properties of those slip planes, and so you can sort of see where one of those slip planes sits in this system. And it's not entirely obvious from that view, to be honest, but what we can see is that we can look at that internal surface and start to rationalize the roughness of that surface, look at the directionality of the movement of the molecules as we compress them, and we can use that to start to really drill down in a structural understanding of what's going on in these systems. And so, to wrap up this, I want to think about the digital future and what that might mean. And I'm riffing on this a little bit, and I'm not entirely convinced by it myself just yet. So we're on a little bit of shaky ground. But if we wanted to think about how we could apply these and how we could be really predictive in terms of digital transformation in the pharmaceutical industry, we might want to do something along the lines of this. So we've got our new molecule, right? And we want to know how that's going to behave in the solid state, so we can start to think about developing it, we can start to think about processing it, manufacturing it, right? So what we can do is we can use all the information in the CSD. So I talked about Mogul, and I talked about being able to understand bond lengths, bond angles, valence angles, torsions, in terms of the context of the CSD, but we can use that backwards as well. We can be predictive about it, and so we can use all of that geometric information to form very plausible, highly probable solid state conformations of a molecule, right? So that's what we've done here. We've gone from a 2D diagram, and using information in the CSD, we can generate a 3D conformation of it, right? That's the simple bit. The next bit is your black box crystal structure prediction. Claire Adjman's in the audience, I think, so speak to her if you want to actually know anything about that. But this is becoming more routine in the pharmaceutical industry, right? These approaches, these sort of what were really complicated, expensive, high-end computational approaches to think about going from a 3D molecule, a 3D structural molecule to a crystal structure, they're becoming more tractable, right? They're still expensive, they're still time-consuming, but we're doing them more and more and more, and that's what we're seeing across the pharmaceutical industry, right? And so if you do a good crystal structure prediction, you get lots of potential answers to lots of potential different questions. So you form all these different crystal structures, right? But what can we tell about them? And can we use them to understand processes a little bit more, understand the behavior of different systems in a little bit more detail? Are there some things that we know are always going to form needles? Are things going to be sticky and we don't want them to be sticky? Or can we have systems that are going to tablet quite nicely that we can predict from these? And so I want to think about morphologies a little bit to finish this off. And so we can think about this concept of the morphology landscape. And we're starting to do this with the CSD theory stuff, not quite in this way. This was a bit more of a sort of experiment doing this sort of thing. But we can come up with an idea for a morphology landscape. And some of you will be familiar with this idea of a Zing plot. So that's when we sort of take our morphology that we either have experimentally or that we've predicted, and we sort of calculate relative ratios of different edges of the bounding box that we put around that. So if we sort of look at the small to medium ratio of that box, and we look at the medium to long ratio of that box, and we sort of plot out those different ratios, we can find the 4 different shapes that crystals ever exhibit. So we've got our needles and our laths, which we don't really want. We've got our platey things we don't really want either. And we've got our blocky things, and that's where we want to be, right? Up in here, because then good things happen and pharmaceutical people are happy. And we can do this crystal structure prediction, right? So if we do this crystal structure prediction, we can understand the relative energies of the different polymorphs, the different solid forms that might be accessible to us. And if we can do a morphology prediction, we can calculate morphologies for all of them, right? And so we can construct a chart that looks something like this. And if I put these lines on it to guide the eye a little bit, we'll see that this is for mebendazole. You can see that we're going to tend to form laths and needles for this. If we look at our landscape, we can sort of see for all of our lower energy structures, for all the things we're probably going to see, we're in this bit here, right? And that's probably not where we want to be, but that's a little bit of process understanding that we can get from this. And if we compare that to experiment, this is quite nice because it actually worked. If we look at more bendazole as we grow it from crystals, it tends to form these sort of needly lathy type things. These aren't so needly lathies, but those have been achieved through very, very careful solvent control. And so it's not telling us everything, but this approach, if we're doing the CSP anyway, and we've got good enough models to go with it, then it might tell us a little bit about what we want to know. And so can we actually use this approach to go from a molecular diagram to think about understanding process behavior? That'd be great if we could. But if we do know the answer, how can we control this landscape? Right? If I want to be up there, how can I actually make sure I'm up there? What does that mean in terms of the stability of my solid form? Is it even achievable? And so to wrap up, how do we get the future that we want? There's lots of different ways that we need to take this, but the future is built on data. I'm from the CCDC, right? I'm going to tell you that the future is built on data, but it is by speaking to people in this room, by speaking to all of our partners, we understand that. But it's what other data can we put alongside it, right? Having the 3D structure, having the crystal structure, isn't often enough anymore to answer some of the questions that we have. And so we need to know physical properties alongside these crystal structures. How we link that is very challenging. But we want to know how crystals came to be. You know, this sort of feeds into the CCS that's getting developed here. Being able to understand that is really important. Incredibly important to this approach, though, is negative data. We can't train more advanced machine learning models if we don't know what doesn't happen. And the CSD, it's a wonderful resource, right? But it's a database of positive outcomes, right? I couldn't tell you what experiments made a crystal not happen because they're not there, right? So we need that negative data as well. And so that data connection pieces is really, really key. And actually being able to connect the data to the sample, to the structure is absolutely crucial. There's a lot of activity goes into literature mining of data, and that's really good. That's a really valuable exercise, but it's challenging as well. And so we want to get to a place, and we are getting to that place really, where we can sort of record those properties that we're interested in with very well-defined standards at the start. And those data standards are really, really critical as well. I think we're lucky at the CCDC, we're lucky with the CSD that data standards in crystallography were established very, very early on. So when that graph at the start was really still quite small, if we were trying to do it now, it'd probably be an impossible task. And so this needs to happen across pharmaceutical development and manufacturing, and it is. And we continue to do that by keeping on doing what we're doing. So the collaborations that CCDC has, the collaborations that you have with colleagues and other people in this room, pushes us along in that journey there. Because these collaborations provide real systems, right? We can do as much sort of navel gazing on sort of CSD structures. I can tell you everything you might want to know about paracetamol, right? But unless we actually collaborate, and we collaborate openly and honestly, we're never really going to get those real systems. And they provide real understanding of development, manufacturing, and processes. And we need to be able to have these cross-industry data standards, because that means that not only do you get industrial uptake, right? You've already had you said what you wanted to say about it, and so you've got those data standards established by you. That means that uptake's easier, but it also provides a platform for really clever, fundamental data science academic research, and that's really, really crucial for this bit of digital transformation. And so, finally, I did very little of this myself, to be honest. A number of people to thank. Lots of colleagues at the CCDC, particularly in the solid form and particle science teams. And Ian, who some of you might know, he's quite involved in the DM Squared project, and he very kindly challenged me on the way that I finished this talk, and I think it was probably better for it. Lots of friends and colleagues at CMAC, particularly Laura and Julia and the groups that supported them. Various collaborators elsewhere. Members of the CFC, Adopt, DDAP, lots of people that funded it, and yourselves for paying attention to me. Thank you very much.
Thank you very much, Andy. Sorry, I'm one-handed on the mic. I need to speak into one. You can get your own. There's a queue. So we've got a question on the app. Well done, first one of the day. We've broken the duck. Prize goes to Peter. How do we chase negative data when other objectives often hinder time being spent on making it public?
So that's an excellent question, and I think how you chase negative data in the pharmaceutical industry is a challenge. And I think I hope that by thinking about negative data as not being a bad thing, and with sort of the increasing use of lab notebooks and really recording everything that happens, that feeds into that negative data. But what that doesn't do is really get that outside, and I think that's where it needs to be. And so we often talk about removing the stigma from publishing negative data sets. And it should be absolutely fine to be able to write a paper and say, I tried all of this, none of it worked. Because these are data points, these are important data points. Just because you don't have a fancy story to tell at the end of it about your hypothesis that was, funnily enough, proven by the results that you got, being able to actually actively demonstrate those negative points is huge. And so I think there's work we need to do as a community, at the CCDC, working with publishers to encourage that as well.
You see, I mean, that's just the practicalities of it. I think the question's touching, there's a cost associated with this. So I think the will is there of all the projects that maybe, focusing on negative results actually is you're convincing yourself of the learning benefit of that. But, acquiring your data from all your experiments, putting the time and effort to write them up, make them in the form that are shareable. Is that something in your discussions that's also, part of the discussion beyond saying this is a good thing to do, just the practicalities?
Yeah, so I think that's always going to be a challenge, because we all want to get the results, because results mean papers mean prizes and PhDs, right? But I think sort of having to take that pragmatic step, and I think some of the ways that we're advancing with other digital aspects of things that enable that to happen more quickly. If we can start to automate more of that process, if we can sort of dial in, this is the experiment, these were the conditions, this was the outcome, bang. And keep it like that. I think digitalization is going to help. So being able to automate that, though, takes a little bit away of that onus on somebody having to record that, and that makes it a little bit cheaper.
Maybe setting up some of the discussions in the coming days, perhaps. Any other questions for Andy from the audience? Van. Special.
Thanks for the excellent talk. So I was just wondering, obviously a lot of, you might, I think about a crystal structure as almost a thermodynamic endpoint. Are you doing anything to augment your predictions, taking into account some of the kinetics, for example, attached to crystallization, just so that we can become better at predicting some of the outcomes, for example, from a polymorph perspective?
So understanding sort of the relative stabilities of different polymorphs in the database and how we can use that information to feed into these things is an interesting approach. Now, there's nothing that we're actively doing in that place just now, because having an idea about sort of the kinetics behind the system, that's one of the things that we have at the CSD. We've got these sort of snapshots of all these experiments. An interesting question that I sort of lie awake and think about at night is the CSD a thermodynamic or a kinetic database? I don't know the answer to that question. I think it's probably more of a thermodynamic database, but it might not be. So the first structure in the CSD, AABHTZ, right, that's from the 1970s, right? I don't know if that's got another polymorph because nobody's looked at it. But I think there's other ways that we can think about the data that we might have, and we might be able to put that into the model. So by thinking about some of the additional data about displacement parameters that we have, so when do different hydrogen bonds form, how do the geometries of those hydrogen bonds vary based upon temperature factors, for instance, is an interesting way to approach thinking about it. I don't know if we've quite got the data to really answer the question that you're asking, but I think that there's ways that we can think about it in smarter ways.
While you're all thinking of your questions, I'll come to you, John, in 1 second. You mentioned standards. I was involved in some discussions yesterday, and people were last week. People were bemoaning, just how long that process takes, and so proposing all sorts of ways of trying to impose standards and so on. It's a challenging, area. Have you, in the context that you've raised, standardising, approaches around data, for example? Do you have any thoughts on how that can be achieved?
So I think you've got to be pragmatic, and I think it's got to come from the entire community. It can't just be somebody saying, do this and it won't work. And so what's quite interesting is you can sort of see the evolution of the CIF file, right? So the crystallographic information format file that was sort of proposed in the early 90s, and you can sort of see the way the CSD changed and the way that graph changes upon when the CIF happened and started to exist. The key thing that the driver was that there was instrument manufacturers and software providers that for crystal structure solution and refinement adopting the CIF as that standard. And they adopted that because I think there was a good bit of foresight there as well, but a lot of it was because they had a stake in the game, they were part of the conversation, and Frank Allen of CCDC was a very persuasive man, I think. So you need a bit of carrot and a bit of stick, I think, for these things, but you need to make that journey easy, and so you need to get everybody involved. If you can convince instrument manufacturers that it's the right thing to do with a sort of established set of standards that you want, and you can get them to adhere to it, then it just sort of happens a bit more naturally.
John?
You did a very good job stealing my question.
I hope one more.
Yes, my question was coming back to the slit planes. So if I've understood rightly from the images, it's more of almost a physical, do you have teeth or is it a smooth plane? Is there anything actually where you've looked at sort of the interactions, so the strength of the interactions between the planes as well, or is it purely a physical topography effect?
How in depth do you want me to go in this answer? So
I'll let Alistair answer that one.
So there's an awful lot more going on there than really just sort of looking at a crystal structure and going, yeah, that's got a gap in it. So we do think about lots of different descriptors around those slit planes as well. And so I didn't cover them in the slides today, but we do think about the nature of the interactions across those. So if you've got hydrogen bonding, if you've got your intermolecular interaction glue that's holding those together, that's going to be an impediment to slit, rather than if you just got sort of van der Waals forces that happen. The actual separation of the planes is important too. So if you've got sort of a tiny little slit plane that's like a hair's breadth thin, that's going to be different if you've got like a bigger gap between them. The relative distance between the planes, so what is your crystallographic d-spacing essentially? But the distance between equivalent planes is important as well, and so you can sort of think about that. If you've got a stack of sheets of paper, and you've got a stack of sheets of thicker card, if you try to slide them over each other, the sheets of card will sort of slide more uniformly and easily over each other because the relative distance between the equivalent planes is simpler. And we also take into account the relative orientations of planes. If you have two potential slip planes that are around about 90 degrees to each other, you get really interesting mechanical properties. That's where you get the sort of bendy crystals that you can tie in knots that you sort of see in the literature quite a lot in there. So there is a lot goes into it, and more than that. There's a lot of really sort of physics, materials physics from like the 1950s that goes into it as well. So Burger's vectors and line energies and stuff, but that's too much for 5:15 on a Tuesday.
Thanks, Andy. Any final questions? If not, if you join me in just thanking Andy for a great presentation.